Hello, Biology 102. Today we are talking about the chemistry of DNA. DNA we really didn't know much about until the 1860s when Gregor Mendel began his studies of inheritance. And Gregor Mendel gave us some laws of inheritance and talked about a chemical that was being passed from parents to offspring. It wasn't until the turn of the century, so the early 1900s to mid-century, that scientists started to really hone in on this chemical and start to test it, start to discover what chemicals made up this inheritance material. They knew that it was kind of where it was found in the nucleus. And by the 1950s, the world was convinced that DNA was this material that was passing from parents to offspring and influencing inheritance. And people knew this particular chemical, but they didn't know exactly what its overall chemical structure and shape looked like. And so there was almost like a race to figure out the double helix. In 1953, we give Watson, Crick, Wilkins, and Franklin credit for figuring out that this DNA was actually this like kind of helical shape and the overall structure of DNA. So today I want to share with you a little bit about the chemistry of DNA and how this molecule looks and behaves inside of us. So we have talked about our 4 macromolecules, and one of them is a nucleic acid. Nucleic acids have a monomer and a polymer like every other macromolecule that we have talked about. For nucleic acids like DNA and RNA, the monomer is a nucleotide. And a nucleotide is almost like a letter L shaped chemical with a sugar in the middle here, a phosphate group, and a nitrogen base. And so it makes this kind of letter L shape. If you look at my picture over here, in yellow is our phosphate group. This house-shaped blue structure is the sugar, and then you have this nitrogen base with these nitrogen groups out here in green. This is almost like a pearl on a necklace that is going to repeat over and over again to build the polymers of DNA, which is deoxyribonucleic acid. That is a double-stranded, so imagine 2 letter L's, almost like a ladder, that build this particular structure. And RNA, ribonucleic acid, is also a polymer of this kind of class of macromolecules, but it is single-stranded. So imagine your ladder cut in half so that you only have 1 letter L that's kind of repeating down one side. So I want to talk a little bit about this monomer and how it repeats, first in DNA, and then we'll look a little bit at comparing DNA to RNA. So what does a double helix look like? In terms of DNA, you have along the sides of your ladder, you have a phosphate and a sugar group, right, that repeat. And you can see it in my structure over here. You have the yellow phosphate repeating with the blue sugar. So phosphate, sugar, phosphate, sugar. That makes up the sides of the ladder. And then inside, the rungs of the ladder are these nitrogen bases. These nitrogen bases come in 4 different kinds or types for DNA: adenine, thymine, guanine, and cytosine. So A, T's, and G's, and C's that you see internally here. Now, these phosphate and sugars along the sides are covalently bonded. They are quite strong in terms of their bonds. The inside rungs of the ladder that you would step on, right, the nitrogen bases, have a hydrogen bond, which is a weaker bond. This is important because when we copy our chromosomes in before cell division, like mitosis, meiosis, when we're making new cells, we need to copy all of our chromosomes. And so we need to be able to pull apart DNA, break the weaker hydrogen bond, but keep the code consistent, because any changes in our DNA code makes a mutation, right? And mutations are often harmful for us. And so having this strong outside bond that holds this letter code, right? So these are covalently strongly bonded, holding this code in place so that it doesn't mutate, but still being able to separate the 2 sides of DNA, then either read for a gene to make a protein inside of our bodies, or even copy the chromosomes before cell division and reproduction, right? And then being able to snap this molecule back together. That hydrogen bond is weaker, so we can break it, but then it also like kind of comes back together very evenly at the end of these processes. So this difference in chemical bonding along the sides and the internal rungs is really important for how DNA is going to function. Now, when we look at these nitrogen bases, we have 2 groups. Purines are double rings. You can see they are a larger chemical width with, you might say. This includes adenine and guanine. And then over here, we have the pyrimidines, which are a single ring. That includes thymine and cytosine. And if you remember, A's always bond with T's. So in our center rungs, adenine will always bond with thymine and guanine with cytosine. When I learned this in chemistry and in high school, I grew up in a small town where corn in the Midwest grew in the center and the outskirts of our town. So I learned this as at town corn grows. That was my mnemonic device to remember these. So come up with some way to remember that A's, adenine, always bond with thymine and guanine with cytosine. But the interesting thing about this is it's always one purine with one pyrimidine. And the reason is because DNA is not a lumpy molecule. By putting one purine, a double ring with one pyrimidine, a single ring, you have sort of an equal chemical width along those rungs. So it's not a lumpy molecule, but it has kind of a fixed chemical distance inside of there because you're having a double ring, always with a smaller single ring, A with a T and a C with a G, so that we have that like sort of conserved chemical distance within our ladder. So this nitrogen-based pairing is important, always a purine with a pyrimidine to kind of keep that very consistent inside of our structure. The last thing I want to mention about DNA is that we say that DNA is anti-parallel. It is anti-parallel because the two strands run in opposite chemical run opposite of each other, but in terms of chemistry. And so it's almost like you have one letter L and then the next one is like the one that is attached to it would be, rather than like I've been showing, it's almost like it's upside down and then that repeats over. DNA is anti-parallel because the two strands run in opposite directions, and the chemical directions are determined by the carbons and the naming system, and whether you end essentially with a phosphate group or a sugar and a nitrogen base sort of connection. The phosphate, it turns out, is off the fifth carbon, so we say that it is the 5' end of DNA. So here's your phosphate group that's running on one side 5' to the sugar, which is the 3' end, this sugar connected with the nitrogen base. So one side will run phosphate to sugar, or 5' to 3', and then the other strand will run opposite of that. It will run from the sugar, the 3' end, to the 5', or this phosphate circle. And so one side runs 5' to 3', the other side 3' to 5'. DNA, is called a double helix. And so this kind of chemical bonding and the way that this is set up allows DNA to have enough, kind of a fourth of a degree turn, almost like a spiraling staircase, and it gives it that helical turn that we see in this molecule and that people know sort of about DNA. The last thing I want to do is just kind of tell you a little bit about how RNA is different from DNA. So RNA is the other kind of nucleic acid that we're going to talk about. RNA is single-stranded, so like DNA is a ladder. If you would cut that ladder in half, then you would have a single-stranded RNA. So it is typically smaller, not only by width, but also RNA is often not as long as DNA as well. It's typically representing a single gene or a smaller portion of DNA, or something smaller than that even. And so RNA is often smaller. It can often it's so small that it can exit the nuclear pores, whereas DNA is big enough that it gets stuck in the nucleus. And so RNA has more kind of free reign in the cell, and it can often kind of go between the nucleus, or some types can go between the nucleus and then to the outside cytoplasm and things like that. And we'll talk about that a little bit later in protein synthesis. The other differences are in the chemistry. So in the nitrogen bases, you have A's with T's and C's with G's and DNA. And in RNA, we have A's with U's, or uracil. So thymine gets replaced with uracil. C's and G's are still together. But that U is a different sort of nitrogen-based possibility in RNA, and then thymine disappears. The sugar is also different. Ribose sugar is used, so this is why it's called ribonucleic acid. Deoxyribose is what is used in DNA. That's why we call it deoxyribonucleic acid. But ribonucleic acid uses this ribose sugar, which has an oxygen, like some oxygen that DNA doesn't. Deoxy means you're lacking some of those oxygen, a part of that like particular molecule. And lastly, while DNA kind of has one form that is housed in our nucleus as our code, RNA comes in a variety of different forms. We're going to talk about 3 different kinds that work for protein synthesis at the end of this unit. So messenger RNA, mRNA, rRNA is ribosomal RNA, and tRNA, transfer RNA. And so we'll be talking about those kinds of RNA. So I hope this gives you some information and chemical sort of knowledge about DNA and RNA. I will see you next time.